Our first speaker is Dr. Tracy Bale. She is a professor of neuroscience in the animal biology department in the veterinary school and in the Department of Psychiatry in the School of Medicine. She received her PhD in pharmacology and neurobiology from the University of Washington in Seattle, and her postdoctoral training at the Salk Institute in California was focused on stress and hormones. And her lab at Penn is specifically interested in understanding the role of stress dysregulation in neurodevelopmental and neuropsychiatric disorders, with a specific focus on sex differences. Recent studies from her lab have identified novel mechanisms involved in the effects of stress on multiple generations of offspring. Tracy has received numerous prestigious awards, numerous prestigious awards, including the Medtronic Award from the Society for Women's Health Research for outstanding research that has led to the improvement of women's health. So without further ado, Dr. Tracy Bale.
Thank you, Leo, and welcome. So with all that serious science now aside, we're going to talk about some more entertaining aspects of the research that we do in my lab. And we're going to focus today on one aspect that's near and dear to my heart, which is blaming your parents. And that's because I am the mother of a teenager, and I know that I'm constantly blamed for everything. So let's start off with some humor. So 3 scientists walk into a bar. They notice 2 alcoholic mice, a mother and her son, sitting on bar stools, madly sipping up gin from thimbles. The mother mouse looks up and says to the 3 scientists, "Hey, geniuses, tell me how it is that my son got into this sorry state." "Bad genes," says Mendel. "No, it was your helicopter parenting," says Freud. "Actually, it was your ancestors' unfitness," says Darwin. As the 3 continue to argue, in walks a very smartly dressed woman who settles their debate. Clearly, it was not enough exercise or White House garden vegetables. So we all know that, and it's not surprising, that early development, if you think about a baby's, if you think about a baby's early development, and you would think about the environment and how it might influence or shape, and as Leo pointed out, that plasticity involved in early development and how the brain will function later on. But what might be surprising to you is actually to think about the fact that both male and female, so mom and dad's life experiences could ultimately shape and program germ cells that would contribute then to that next generation and possibly future generations. So we've learned a lot from Gregor Mendel sitting in his pea patch. We know a lot about genetics. You guys all know this. You know that genetics are the backbone of your DNA, ultimately shapes your development and who you are. But what Gregor Mendel couldn't explain is how the environment itself could future program that offspring or future program the peas in terms of the influence of the environment in an epigenetic way. Evolution does account, however, for influences of the environment, obviously, but it moves glacially slow. And Darwin really couldn't explain the dynamic aspects of the environment either. So neither Gregor Mendel nor Darwin could explain the influence of our ongoing changes in the environment, especially with the current epidemic of obesity. So how is it then that we can think about the influence of the environment on these aspects of brain development? Was Lamarck totally wrong? Is it possible that the influence during pregnancy or even prior to gestation, the behaviors that we have and the influences could ultimately shape our offspring? And perhaps Lamarck wasn't so crazy after all. We now know a lot about epigenetics. We know that the influence of the environment can program gene expression through multiple generations. But what is epigenetics? Epigenetics is simply the modifications of DNA that don't alter the sequence of DNA. So that can be found in terms of histone modifications wrapped around nucleosomes, methylation of DNA, or the increase in expression of small non-coding RNAs. And that's very simply what epigenetics is. So let's talk about maternal exposures first. And by maternal exposures, we know that it may be such things as infection, maternal obesity, maternal dietary changes, maternal stress, etc. And those can occur prior to pregnancy, over the course of pregnancy, or at parturition. But how those influences develop and shape the developing brain are in large part in relationship to how the milieu or the maternal environment, the maternal hormonal environment, has access at the placenta and through the placenta to ultimately shape that first generation of somatic cells, including the developing brain. Not surprising. But what might be surprising to you is to know that the primordial germ cells that will give rise to that second generation are also present and can be exposed during this initial environment. Therefore, in other words, whatever grandma ate or was exposed to or did during her pregnancy could not only affect her kids, but her grandkids as well. So we've actually used in the lab a model of maternal stress. We exposed female mice during early pregnancy to stress, and we studied their offspring. And we found that these male offspring specifically show significant changes in the first and into the second generation, whereby they show changes in post-adolescent growth and development, hyperreactivity of their stress response, and diminished cognitive abilities. And this is in the case of just males were showing this effect and not females. So there's a sex bias here, which is interesting when you think about neurodevelopmental disorders like autism and schizophrenia. So how is it that mom's exposures and experiences during pregnancy shape and affect the developing brain? Well, we think in large part these have to do with the development of the placenta. And we've used that information in our mouse models to be able to explore relevant and important mechanisms that ultimately shape the developing brain and can impact, as I said, first and second generation offspring. And one way that we can do this, and the reason that we use mice, is called transgenics. We're able to manipulate DNA and ask very specific questions in mice, the relevance of those genes to shaping the developing brain. Here I'm showing you an example of a placenta in an animal. This is a developing fetal mouse, and this is the placenta. And what we can do is we can use transgenics to specifically target genes within that placenta and ask questions about what genes are relevant for the developing brain and respond to that environment. And in our studies in the lab, we identified one specific gene, OGT, or O-glycosyltransferase. This enzyme is really important for the function of the placenta, and it sort of acts as a canary in the gestational coal mine. It's constantly sampling the maternal milieu, and it reads that and changes the function of the placenta, and then imparts that information on the developing fetus in order to impart a fitness for what the environment is expected to be when that offspring grows up. So then we can ask the question now, okay, we think this gene is important in the placenta. Now show me, how do we decide that this gene in the placenta is actually important for the developing brain? We go back to transgenics. We can target this specific gene just in the placenta and then ask the question, can we recapitulate what we know to be true about stressing mom and that offspring phenotype? Can we recapitulate that by targeting this one gene? And the answer is yes, in fact, we do. In fact, in the male offspring and female offspring, in which we've only manipulated one specific gene that we identified in the placenta, just one gene, we can show exactly the same phenotype, whereby there's a post-adolescent change in body growth and development in both male and female offspring, as illustrated here, as well as dramatic changes in gene expression in those brains. This is at postnatal day 2, which is right after they've been born, they've developed a bit. These are control animals. It's a heat map. This is animals that have been exposed to reduced OGT in that placenta I told you about. And what you need to know is that the gene expression patterns, where they're low are high in the OGT animals, and where they're high in the control are low, meaning that what is supposed to be happening in normal growth and development in that brain is completely changed in those offspring because we manipulated one gene in that placenta. So what is another mechanism by which mom's stress experience can program the developing brain? I'm sure you've all heard about the microbiome. Well, we've begun on a study recently in the lab thinking about how the maternal vaginal microbiome can shape and affect the developing brain. Because it might be surprising to you to know that as the baby is born and travels down that birth canal, the baby gets a mouthful of the mom's vaginal microbiome. And that's important because that microbiome populates the baby's gut microbiota and ultimately shapes the function of that gut microbiota as well as its metabolites and absorption of really important nutrients important for brain development. So you all know, you've heard this in the media, you know that the microbiota are plentiful. It's the most abundant fixture of over trillions of microbiota in your body. We know that this is really important for shaping the function of your gut as well as interactions with your immune system. This is ultimately important because it is shaped large part during early development. Your interactions and exposures to germs shape not only your immunity, but also how your gut microbiota for life will respond to that changing environment. During early development, we know that the microbiota are important for how nutrients are broken down and absorbed, and this is really important for brain development. So in our maternal stress model, we looked at this, and we found, in fact, this to be true, that if you stress mom early in pregnancy, you can change by reducing these plentiful and abundant and positive, meaning beneficial microbiota in her vagina. And ultimately, you see the same response in her offspring's gut. And if you actually look at this relationship whereby stress is changing or reducing those beneficial organisms, it's a one-to-one relationship, meaning the changes you see in that offspring's gut are predicted from the changes you saw in mom's vagina. And again, this is really important because if you reduce the beneficial, positive, or influential bacteria, you can change which nutrients are broken down, absorbed, and how it affects the brain. Okay, so now we've ultimately thought a lot about mom and her influences, whether at the level of the placenta or the level of her vaginal microbiome and shaping baby's brain. But what about dad? Can't we blame dad too? Ultimately, isn't dad supposed to be responsible as well? The answer is yes, of course we can. But the beautiful thing about studying rodents is that one of the difficulties we have in the lab and looking at epigenetic transgenerational effects in females is that we have many things to contend with, right? It's just not as simple as her germ cell contributing to that offspring, right? You have pregnancy in the uterus, et cetera. So in the female mouse, during development, you have to contend with her uterus, her pregnancy complications, her parturition complications, lactational deficiencies, and her behavior that shape the developing offspring brain. But in male mice, there's one cell, only one cell that you have to know about. Ultimately, if dad is exposed to something throughout his lifespan, and he's able to pass on a phenotype to his offspring in a mouse world, he is only around to contribute one cell that we have to contend with. And so we know that if that mark is present there, we should be able to isolate that mark and determine its importance. And so we did that in the lab, in fact. We took male mice, we exposed them to a period of stress, either over puberty or as adults. And after this period of stress, we gave them some time off so they're no longer stressed out animals. But if that stress were to impart a mark, then we should be able to breed those animals and look at its offspring. And sure enough, what we found in the lab was both male offspring and female offspring, no matter when that stress occurred, imparted a difference in how his offspring reacted to stress when they were adults, even though those animals had never met their dads. Pretty impressive. What's more impressive is thinking about, well, now we know that that mark had to be present in those germ cells. That sperm had to impart a message at fertilization. So what is it? What epigenetic mark would we look for in all of this complex mixture? Well, we started in the lab by looking at those non-coding microRNAs. And we did, in fact, identify 9 of them that were increased in both stress groups. So now you would ask yourselves, well, how do you know that those 9 microRNAs that you're associating, right? This is a correlation. They're increased, but how do you know that they're causative? How do you know that they impart an effect? Well, the way that we do that is we take those 9 microRNAs, we take a fertilized egg, and we inject them right in there and ask, do those increased 9 microRNAs make any difference in those offspring? We give them to surrogate moms, we raise them up as adults, and then we look at their stress response. And has it been changed in the same direction? And in fact, it was. Amazingly so. So we injected those 9 microRNAs. That's the only difference we did. And we made the exact same phenotype in these offspring. Both male and female offspring show that same change in their brain development and how they respond to stress. So now we know that those 9 microRNAs are incredibly important. And when we look in the brain of these offspring, as you can see, here's the control offspring and the ones that had injections, night and day of how their brains developed following those 9 microRNAs that were injected way back in the zygote. So clearly, we are imparting a major effect on how those brains are developing. Why is this cool? Well, this is really cool because now you can think about, well, maybe we can be screening as a biomarker human sperm because it's pretty accessible. And we can actually ask the question, could we find marks in human sperm that may be predictive of risk or resilience for future offspring, depending on dad's experience and exposures? So ultimately, what this leaves us with is, why does your brain respond to stress in the way that it does? And it's really complicated, yes. And much of what we thought we knew is really gone up in smoke in terms of just related to our genetics. We clearly understand a lot more about the epigenetics, the lifetime, the lifespan of things that you've been exposed to may shape your offspring's brain. But what I will leave you with is that no matter what, I think it's pretty clear we can still blame our parents for all of our woes. So thank you very much for listening and coming out tonight. I especially want to thank members of my lab who've done all of the work that I've talked to you about tonight, especially Chris Howerton, who did all the placental work with OGT story, and Allie Rogers, who is actually here tonight, who did all of the work on the sperm story, which is very exciting, and of course all of our funding agencies. So thank you.
Anybody have any questions for Tracy? We'd be happy to take them now.
We'd be delighted.
Pick up the mic.
Just shout.
Anybody? Mic's on the side.
Sorry.
My question is in regard to the effects of the maternal vagina microbiome on the offspring brain development. So what differences do you see in individuals born by cesarean section?
That's a fantastic question. So actually there's strong evidence that by not getting the vaginal microbiome, that those guts are set up differently, the interaction with the innate immunity is different, and thereby those children are at greater risk and do show higher levels of things like allergies, immunity issues. So there are clinical trials going on, some in this country, a lot in Canada and Europe, where they actually do take vaginal lavages and expose cesarean delivered babies so that they do get that. It's a great question.
So is there, this is, I can't recollect what study it was, but there's this assumption that kids who grew up during World War II, like in Europe, who experienced stress and starvation, their children are more healthy than...
So this is the Dutch hunger winter. So there's many different epidemiological studies that have been done, the Swedish famine, the Dutch hunger winter. Yeah.
But your findings are that it might have a negative impact on the brain, but...
You'll notice I didn't make that assumption at all. So especially with mice, we just know we can measure their stress response, right? We have a physiological endpoint. We can say that it's different. All that I've really told you is that things that we've done to mom or to dad can change the development of the brain. Now, whether that's negative or positive would probably depend a large part on the environment that they're living in. So the offspring that I showed you, in a lot of cases, sometimes they're more stress reactive and sometimes they're less stress reactive. I can't really put that as subjectively better or worse. And the Dutch hunger winter data actually had both outcomes, right? So it just depended on when they were exposed and if it was male or female, multiple generations. In some cases, they had better longevity. In some cases, they had more cardiovascular disease. So it probably has windows of susceptibility, right? When you were exposed determines a lot about your health consequences. And then, of course, the environment that you live in.
Do you have any idea of the, oh, sorry.
Go ahead.
I'll go. Do you have any idea of the mechanism that the RNA actually
Where
is?
are you? Can't see you. Wave your
I'm over here.
Oh, sorry. I was looking for you back there.
But like I said, do you have any idea of the mechanism that the RNA is using to change things from?
In the sperm studies?
Yeah.
Another fabulous question. So
Thank you.
Allie's paper is right now under review. She did a fabulous study where she actually repeated the injections. And we think that the microRNAs were targeting the stored maternal mRNAs, which are present in the oocyte. So she actually did that amplification study. So she basically re-injected those microRNAs, took what is essentially a single cell, which is amazing, right? A single cell, and did an amplification of the RNAs present, and found, in fact, that the microRNAs are targeting the expected stored maternal mRNAs. So the microRNAs function to degrade mRNAs. And so they seem to be functioning in that state to degrade those mRNAs.
Thank you.
Did you look at any other epigenetic modification rather than microRNAs?
So we have not looked at any other modification. So Chris Pierce, I know, who is here tonight and spoke in the previous session about the cocaine model, has looked at some other marks of methylation and histone marks. Other paternal models, similarly in rodents, have looked at like the methylome differences. We have not in our model. The reason we chose to look at the microRNAs was because we could do this manipulation and ask if those microRNAs were doing anything. We still haven't looked at the histone marks, but there are retained histones, some in sperm, and there certainly is methylation we could look at as well.